In this video, we will discuss about when we knock out this NPY1r in the pancreatic cancer, in the pancreas only, then what is its impact on the metastasis of the pancreatic cancer. In the previous video, we have understood that in the highly metastatic pancreatic cancer models, there is a upregulation of not only of this NPY protein but of this NPY1r or NPY receptors. And so in this video, the objective is to check the metastasis of this pancreatic cancer to the liver, as liver is the most common organ of the migrations of this pancreatic cancer before it migrate to the other organs. So they have developed this pancreatic-specific NPY1 knockout, and there are three different cases that they checked. First one is in which this NPY1r is present as it is, or we can say that wild type conditions, and is in the other one is the NPY1 is knockout in one of the allele, and in the third conditions where NPY1 is knocked out in both the alleles, or we can say the homogeneous NPY1 are knocked out and heterogeneous NPY1 knockout. Heterogeneous in which one of the allele is knocked out, and homozygous means both the alleles of this NPY1 is knocked out. So first what they did is they checked the overall survivals rate, and what they are able to find out for all the conditions, even when they compared it with the NPY1 receptors in the wild type pancreatic cancer mice models, they are able to see that there is not much impact of the percent survivals in those different mice models, indicating that this NPY1 receptors does not have too much impact on the patient survival rate. However, when they check the metastasis into the liver, they are able to see that compared to the NPY1 wild type, there is a low metastasis in both the homozygous NPY1 knockout receptors conditions, or second one is the heterozygous NPY1r knockout conditions, and the third one is the homozygous NPY1r knockout conditions. In both the cases, that is homozygous and heterozygous knockout conditions, the metastasis is statistically reduced compared to the case where NPY1 receptors are present as wild type. And the panel D shows the representation image in the first row, where we can see that the metastasis representative image of the liver of the three different conditions, and we can see that in the wild type conditions there is a high presence of these pancreatic cancer cells, and whereas in the case of this heterozygous knockout, the pancreatic cancer cells are present in low amount, and in the case of this homozygous knockout also we see very low presence of these pancreatic cancers. And the immunostainings images are shown in the second row of the panel D, where we can see that the presence of the pancreatic cancers are higher in the liver compared to the conditions where NPY1 is either heterozygously knockout or homozygously knockout. So this indicates that although this NPY1 plays a role towards the metastasis of the pancreatic cancers to the liver, but it does not have any significant impact on the patient, on the survival rate of the mice model. So it is NPY1 could be a useful strategy to curb down the pancreatic cancers or limit the pancreatic cancers within the pancreas, so that they can be easily or surgically removed. In the next video, sorry about this. In the next video, we will discuss what are the changes, what are the observations we will get once we knock out this NPY1 or NPY1 receptors or NPY1r, not the proteins, the receptors, the NPY1 receptors in the whole mouse body. In this case, they did the knockout in a way that when the KRAS NP53 mutations are triggered, then these NPY1 receptors mutations are also triggered. So this is how they did the knockout here. In the next case, we will discuss a mice where in its whole body this NPY1R is knocked down, and when they introduce this pancreatic cancer, then what are the impacts on the metastasis and the survival rate. That's all for this video, and if you need to understand more about this uh, results or experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.